Hello, my name is Hannah Merritt, and today I'm going to be presenting on climate effects on aquatic regeneration of Bohemian knotweed rhizome fragments. My supervisor for this project was Dr. David Clements from Trinity Western University. So to start, I wanted to just explain what Bohemian knotweed is and go a bit more in depth into the details concerning this issue. So knotweed is an invasive perennial plant from the Polygonsae family that originated in Southeast Asian countries like China, Japan, and Korea. Its growth is controlled by adverse environments and native allelopathic plants. However, in 1847, a Dutch doctor by the name of Philip von Siebold brought samples of knotweed to Europe after his time in Japan. And by the beginning of the 20th century, knotweed had spread to Europe, South America, and North America. Knotweed is most prevalent in three forms, as Japanese knotweed, giant knotweed, and the hybrid, Bohemian knotweed, although more than three forms do exist. My research is focused on Bohemian knotweed, a plant that is characterized by the mixed size of leaves it displays. Bohemian knotweed has the unique ability to sexually reproduce, which consequently allows it to produce highly invasive and viable seeds with greater genetic variation. This unique ability differs from the abilities of Japanese and giant knotweed, as Japanese knotweed is unable to produce viable seeds, and giant knotweed produces viable seeds that are not invasive. Keep in mind, however, that the rhizomes, which are the root-like structures of the plant, still have higher regeneration rates than those of the seeds. Additionally, Bohemian knotweed has been identified as the most successful taxon of the Polygonsae family in terms of successful establishment, particularly in North America. Bohemian knotweed is concerning because there's so little known about it. Bohemian knotweed has historically been mistakenly identified as Japanese knotweed, which means that scientists have an extremely limited body of knowledge about the specific mechanisms that allow Bohemian knotweed to grow. Applying Japanese and giant knotweed control methods to Bohemian knotweed is actually not as effective, which again contributes to Bohemian knotweed spread. These characteristics hugely contribute to the invasive nature of the plant. Not only is knotweed highly invasive, but its establishment comes with many serious consequences. Knotweed can alter soil composition by lowering its pH and depleting it of potassium, which consequently limits the biodiversity of the surrounding area. It can additionally limit biodiversity through its allelopathic ability to suppress soil fauna. Beyond environmental impacts, knotweed can considerably impact the economy. Because of its ability to grow through concrete, it can cause serious structural damage. Additionally, the cost of management and control of knotweed is incredibly high, as it can take 2 years to control even one stand of knotweed. For example, in years past, it has cost Britain up to 150 million euros annually to manage and control knotweed. Despite Bohemian knotweed having the ability to reproduce sexually through seed production, the main way knotweed is spread is actually through the regeneration of propagules like rhizome and stem fragments. These fragments are buoyant and are often spread through river or stream systems and deposited on land, where they can regenerate. According to a study conducted in 1995, both rhizome and stem fragments were proven to be far more successful at regenerating than seeds, with rhizome fragments being 2 times more effective than stems at regenerating. Rhizome and stem fragments are able to retain buoyancy for at least a month, with stem fragments sometimes staying buoyant for 2 months. The buoyancy of fragments, therefore, is better than that of the seeds, with seeds remaining buoyant for only 3 to 3.5 days. These properties of knotweed fragments make the plant highly invasive and successful at colonizing different habitats, especially where waterways are available as invasion pathways. An added element to the invasion of Bohemian knotweed is the ever-changing environment presented by climate change. For instance, northern habitats naturally limit knotweed growth because of the late flowering of knotweed plants in September and October, which corresponds with the first frost. As climate change introduces warmer weather, the first frost is delayed, thereby encouraging the northern migration of knotweed. This is further confirmed by a study done by Grunewald et al. 2014, who found that increased temperatures introduced by climate change encouraged the growth of Bohemian knotweed populations in northern Quebec. So I set out to investigate both of the main issues with knotweed, and that is the rhizome regeneration rates in waterways, because that's a main invasion pathway of knotweed, as well as investigating how climate change may impact the regeneration rates of rhizomes. So I did this over 2 summers. I started in 2020, investigating the research question, does immersion in water prior to being placed in soil impact the regeneration growth rates of Bohemian knotweed rhizome fragments? So just very briefly, I wanted to go over the timeline. This study was split into 3 main stages. So once the rhizome fragments were extracted from the 3 sites, 2/3 of these fragments were placed in covered basins of water, and 1/3 of the fragments were placed in soil. The basins of water simulated a wet environment, such as a river, whereas the soil simulated a more dry environment, such as the riverbank, where fragments would be deposited if they were spread through a river system. An additional group of 14 rhizomes were placed in a garbage bag immediately after extraction to simulate a heated environment. So this was just the beginning of testing out the climate change impacts. After 15 days, 1/3 of the rhizome and stem fragments in the water were moved to soil, and the heat-treated fragments were removed from the heated environment. Half of these fragments were then placed in soil, and half of the fragments went into a water basin for 15 days. Finally, the last stage involved the removal of the final 1/3 of the rhizome and stem fragments, as well as the heat-treated rhizomes, from the water to the soil. These were then observed for an additional 15 days, with the total duration of the experiment running for 45 days. So I just very briefly wanted to get into my results of this experiment. I was able to discover that the 15-day water treatment produced the greatest number of regenerating rhizomes. So you can see in these bar graphs over here that I had 3 different groups coming from 3 different environments where I had extracted the rhizome fragments from. And in each group, it did show that the ones in the 15-day water immersion treatment did produce the most rhizomes that regrew. There was no conclusive data on the rate of regeneration from the 3 different sites and the 3 different water immersion treatments, but this was a very conclusive result. Additionally, we did see that the heat experiment did give us some interesting results. So the rhizome fragments that underwent a 16-day heat treatment saw significant growth that was unlike any other rhizome regeneration we had seen in the regular soil planted rhizomes. So you can see over here, the white regrowth came in just 16 days, whereas some rhizomes that were planted in soil and water took much longer to regrow. So mainly because of this interesting regrowth pattern we saw in the rhizome fragments from the first summer, the research question that I investigated over the summer of 2021 was, how does the exposure to different temperatures prior to being immersed in water and planted in soil impact the growth rates of Bohemian knotweed rhizome fragments? So again, very briefly, I just wanted to go over the timeline of my research project during that summer. So at the beginning of the summer, on May 11th, I collected rhizomes and stems, but I will just focus on the rhizomes for now. And I placed the rhizomes in a pre-treatment period where I put some of them in -20°C environment, some of them in a 5-degree environment, some in a 23-degree environment, and then some in a 32-degree environment. So after 2 weeks of this pre-treatment, I did place the rhizomes in water for 2 weeks or 15 days per the results of my previous experiment. And then I finally planted them in soil. And the results of my experiment were quite interesting and very unexpected. So the rhizomes stored at 5°C had statistically higher regeneration rates, so much so, in fact, that the rhizomes stored at other temperatures did not show any signs of regeneration. Now, this was quite unexpected for the rhizomes stored at 32°C because we had seen such an interesting growth pattern the previous summer of rhizome regrowth at, like, we didn't have a defined temperature of what they regrew at, but it was definitely above 30°C. So that was very interesting. One hypothesis we have for this result is that there the rhizome fragments, when they were in that temperature phase, they were not watered. So it could be a matter of them being dehydrated, and that's why they didn't regrow at 32 degrees Celsius. However, further experiments are needed to really determine why we got this result. However, we did also think that the rhizome stored at 5 degrees Celsius had a higher regeneration rate, maybe because of its dormancy and artificial overwintering. So the rhizome fragments might have thought they were in the winter, and then when they were taken out of the 5-degree Celsius environment, they thought it was summer, and then they regrew faster. Finally, just to briefly conclude, from the two experiments, we managed to discover that 15-day immersion period prior to deposition allows for the most regrowth of rhizomes. Additionally, a cold period prior to deposition allows rhizomes to experience an even greater improved growth rate. The next steps for my investigations into knotweed rhizomes would be to investigate climate change and see how it has the potential to increase the spread of knotweed. I would like to thank TWSRA funding and NSERC USRA funding for supporting my experimental research over the past 2 summers. I would also like to thank Trinity Western University and the Trinity Western University Knotweed research team, including Dr. David Clements, PhD, Maria Goncharova, Vanessa Jones, and Aidan Anderson. Thank you.